So our next speaker is George Yaku, who is going to be talking today about identifying the enzymatic substrate of ubiquitin-specific protease 11 in T. al. And his mentor was Dr. Protease.
Thank you for the introduction, Chris. My name is George Yaku, and I worked in the D. Christos lab over the summer. The title of my presentation is Identifying the Enzymatic Substrate of Ubiquitin-Specific Protease 11 in T-cell acute lymphoblastic leukemia. So T-cell acute lymphoblastic leukemia is a cancer of white blood cells. It occurs when a T lymphoblast, which is an immature T-cell, has a mutation and begins rapidly dividing in the bone marrow. This cancer is aggressive because it has the potential for metastasis, because T lymphoblasts can travel into the blood, and then it can invade the central nervous system, for example. So the current treatment for T-ALL is chemotherapy. However, chemotherapy only targets rapidly dividing cells, so it does not necessarily kill stem cells, and there's a risk of relapse. And in fact, 1 in 5 patients will experience chemotherapy resistance. Also, chemotherapy has a lot of long-term side effects, so we need to find better therapies to address both of these problems. So in T-ALL, we've identified that most T-ALLs have Notch1 mutations, and they occur either in the HD domain or in the PES domain. And Notch is overexpressed in T-ALL, meaning that it gains lots of functions to inadvertently activate different growth factors, and it promotes uncontrolled cell growth, and it increases tumor energy uptake, and those are 2 hallmarks of cancer. So our lab has shown that deubiquitinating enzymes interact with Notch1 to promote leukemia cell survival. So a little bit of background, the ubiquitin proteasome system helps regulate protein homeostasis. So in normal cells, we have a proper balance of proteins. And the UPS system can do that through the process of ubiquitination, where it can mark proteins with ubiquitin for degradation. So the UPS marks damaged proteins, misfolded proteins, but also can mark cell cycle regulators, which means the UPS has roles in cell cycle regulation and cell signaling. However, deubiquitinating enzymes can remove ubiquitin, and this will prevent protein degradation. If this happens at the wrong time or at the wrong place, we get a cell that looks like this, where there's an imbalance of protein. So we are looking specifically at ubiquitin-specific proteases, which is just the largest family of deubiquitinating enzymes. And in my work, we used USP11, which has been shown to regulate the cell cycle and cell signaling. It also interacts with USP7, another deubiquitinase which works with the Notch gene to promote leukemia cell survival. So we believe that USP11 could be a therapeutic target in T-ALL. So with that, we performed a preliminary experiment, and the preliminary experiment was mass spectrometry, where we took cells with USP11 and cells without USP11 to identify if protein levels had changed when we knocked out USP11. And we identified that as we decreased USP11 levels through the knockout, we noticed an increase in some mitochondrial proteins. We found 7 significant proteins, and among them ATP5A, ATP5O, and ATP5B, those are part of complex 5. And these are statistically significant. So complex 5 catalyzes the synthesis of adenosine triphosphate, which is where cells get their main source of energy. So it's also exclusively localized in the mitochondria, so we can use complex 5 as a marker of the mitochondria. Our hypothesis was that USP11 is localized in the mitochondria because we noticed that USP11 levels will change the mitochondrial protein levels. We also believe that complex 5 could be an enzymatic substrate of USP11. And we believe this because we noticed that complex 5 levels increase when USP11 was not present in cells. So for our experiments, we used Cuttle-1 cells, which is the cell line for acute lymphoblastic leukemia. In the first part of our procedure, we had to isolate different parts of the cell. So we isolated the mitochondria and the cytosol, which is the soluble portion of the cytoplasm. So we collected 20 million cells, and then we used a mitochondria isolation kit, which basically disrupts the cell membrane, and then it allows us to pellet the mitochondria, and our supernatant is the cytosol. So we collect these samples, and then we had to further lyse the mitochondria, disrupting its membrane, so we could further analyze it with a Western blot. So the Western blot procedure is to take our samples and put them on a gel, and then we ran an electric current through the gel, which allows us to separate the proteins. Once we did that, we transferred the proteins on the gel to a membrane, which we were able to incubate with our antibodies. So we used there are two different antibodies. So the first is the primary antibody, and those are listed below the description. So we used USP11 as our target protein. We used complex 5 as our mitochondrial marker, because it's only in the mitochondria, and we used actin as our cytosol marker. So after we incubated our membrane, we developed with a Western blot. And the results of the Western blot show that USP11 is actually localized in the cytosol in TALL. So if you look at lane, so we have whole cell lane, a mitochondrial lane, and a cytosol lane. We only see a band for USP11 in the cytosol and in the whole cell, but not in the mitochondria. Our complex 5 and actin markers are consistent with these results. But to further validate them, we performed an immunofluorescence assay, which allows us to basically visualize different proteins in the cell. So in green on our left here, we have USP11 stained with an antibody. Then we took the same cells and we stained them with mitotracker. Mitotracker is a red fluorescent dye which will only stain the mitochondria. And then we also stained for the nucleus, and we merged all these three images to get this result on the right. So we're looking for yellow, and wherever there's yellow, that means that USP11 is in the mitochondria. However, we don't see yellow for the most part. But there are two examples here where we see yellow dots. So the yellow dots can be explained in two ways. One of them is that USP11 might be in the mitochondria in very trace amounts. The other could be that one of the limitations of the immunofluorescence study is that we are depicting a 3D cell in a 2D image. So the USP11 and mitochondria could be on top of each other but not inside of each other. So we'd have to perform further analysis to understand why we see these dots. So also we wanted to test whether complex 5 levels are affected by USP11 levels affect complex 5 levels. So we have in lanes 1 and 2 cells with USP11, and in lanes 3 and 4 we have cells without USP11. And we notice that complex 5 levels are roughly the same. This does contradict the mass spec results, although we would have to do further research to understand why we see this. So in conclusion of this work, we've actually proved the opposite of the hypothesis, because we found that USP11 is predominantly localized in the cytosol in TALL. And we also see that USP11 levels do not appear to affect complex 5 levels. And again, we don't know whether it could be an error of the mass spec results or the Western blot, so we'd have to perform further analysis. However, some ways we want to improve this experiment and move it forward is to analyze whether USP11 is functional in the cytosol or whether it loses some function. That allows us to identify whether USP11 has a role as either an oncogene or a tumor suppressor in TALL. Further, we want to know if we can overcome the limitations of immunofluorescence by perhaps using a cytosol marker like actin and repeating the immunofluorescence study. or if we can use cells with USP11 and cells without USP11 to test the specificity of our USP11 antibody to make sure that it's binding to the correct proteins, and then that way we can maybe quantify how much USP11 is in the cell. We also would like to introduce some quantitative data into our analysis, and to do that we could get protein expression data from patients, and we can run a correlation test to further analyze whether there is a positive or negative correlation between USP11 and complex 5. Finally, we're also want to investigate whether complex 5, or whether we could use PRDX1 as an enzymatic substrate of USP11 in place of complex 5. The reason we came up with PRDX1 is because it is an antioxidant enzyme, which means, and we believe that in TALL there might be an imbalance of antioxidants. Such imbalance could cause an increase in ROS levels, which might drive leukemia cell survival. So as a final note, I'd like to thank the principal investigator, Dr. Panagiotis Diakristos, who guided me throughout the way, throughout this project, and hosted me in his lab. I'd also like to thank Dr. Chi Jin, who basically helped me with the everyday aspects of this project and gave me a lot of feedback. And without him, this project would not be possible. I'd also like to thank all the lab members in the Diakristos lab, and finally, Dr. McKinda, Dr. Cuspino, Dr. Lycan, and everyone who's made this program possible.
Do you have any sense of whether, just in a very preliminary way, you might expect the Western blot data versus the spectroscopy data to be more accurate, just to have more relevance or anything?
I believe that the Western blot data might be more accurate because in the mass spec data, we noticed that some proteins decrease at the same amount, and we believe that could be an experimental error. So for example, we have proteins that are decreasing by, let's say, by 10%, and there's maybe 3 or 4 proteins that are doing that. We don't necessarily expect proteins to be all decreasing by the same amount in that respect. So we believe that the Western blot could be more reliable in that sense, but we would have to do further analysis to really determine why. Yes?
John, that was an excellent talk. I forget if you said this already, but the source of TALL cells, is that a cell line, or is this
So we
primary
use
cell
a couple line cell line.
A cell line.
Yeah, so we use the cell line and it's derived from, I believe it's a pediatric male patient.
So I'm just curious if you've considered if you might get some different results if you use cell lines from different patients, so additional TALL
Yeah,
cell lines from different patients?
definitely. I think we looked at mRNA data, and of course this is a protein, we were analyzing proteins in here, but we do see a difference in mRNA data, although we can say whether that translates into proteins. It's not comparable, but yeah, that's an interesting point.
And that was from the same cell line as well, the cells?
Yeah, so Cottle cells versus replication.
Okay, great.
Any other questions?